Hello friends, this is the continuation of Seed Technology Basics Part 5. So once the pollination takes place, next step is fertilization. It's nothing but union of male and female gametes. Here I am giving you the next step after fertilization. What will be the formation of different parts once the fertilization takes place? Zygote will become embryo, ovary will become fruit, ovule will become seed. As you know, ovary is the female part and the outermost covering of the female part where the fertilization takes place. Ovule is the specific place where fertilization exactly happens. This will become seed. Nucellus will become perisperm. Perisperm is the outer covering, and integuments will become the seed coat. New seed policy came in 1988, October 1st. In seed, endosperm is separated from embryo by epithelium layer. The function of epithelium layer is to connect or to separate the endosperm from embryo. Coming to the generalized isolation distance of different crops. Till now we specifically seen different crops. Hybrid tomato, onion and maize for foundation seed 1000 meter and certified seed 500. Different kernel color, foundation seed 600 and certified 300. In sorghum Johansen grass it is 400 and 400, and in hybrids 300 and 200. Loose mut of wheat 150 and 150. Hybrid rice 200 and 100. Mandated crops of ecrisite. As you know, ecrisite works with five crops: sorghum, bajra, then groundnut, pigeon pea, chickpea are the five mandatory crops. Here finger millet is also added. Coming to the minimum number of inspections for certification, I am repeating again, these are the generalization of number of inspections. For all self-pollinated crops, two inspections are sufficient, like rice, soybean, cowpea, wheat, black gram, spinach, barley, green gram, linseed, oat, chickpea, millets, amaranthus, red gram, cotton, sunflower. For cross-pollinated and some of the often cross-pollinated crops like bajra, sesame, chili, turnip, sorghum, hybrid cotton, carrot, rapeseed, brinjal, radish, mustard, okra, tomato, three inspections are required. For hybrids like maize, cauliflower, sorghum, bajra, four inspections are must. Seed drying. Normal drying is practiced in cereals, quick drying in rapeseed and mustard, and slow drying in pulses. Now, protandry condition is observed in maize, cotton, sunflower, sugar beet, protogyny in bajra, nizer, sugarcane, castor. These examples are very important because here it will look easy, but in exam they will be confusing. Protandry is maturation of male parent first. Protogyny is maturation of female first. Okay, means before male part. Syngenesious is having stamens united. It's a condition where stamens are united is called syngenesious. Epipetalous, five stamens are present in sunflower, safflower, nizer. Different kinds of blooming. Basipetal blooming. Basipetal is towards base, means from top to bottom flowering will start in case of rice, sorghum, maize, tasseling. Centripetal is means flowering starts from the middle or center of the panicle, that is observed in nizer, sunflower, safflower, sugarcane and bajra. Acropetal. Acropetal means heading towards upper side, towards the sky, that is found in maize female part, cotton and sesame. Superior ovary is found in rice, wheat, sorghum, maize, groundnut, sesame, castor, linseed, sugarcane and cotton. Inferior ovary is found in sunflower, safflower, nizer. These are just memory based one. Monoecious plants are maize, castor, coconut, whereas dioecious, where male and female parts are present on different plants, are papaya, asparagus, cucumber. Different types of inflorescence. In rice it is panicle, wheat spike, maize cob, sorghum panicle or head, sunflower and safflower head, nizer also head, castor panicle, groundnut gynophore or peg, okra and sesamum capsule, linseed cymose, sugarcane arrow, cotton, dehiscent capsule or ball. Next, we shall see some of the important hybrids or some specific varieties. In wheat, Trupti is HD2833. This is double dwarf variety resistant to all three kinds of rusts. Harshita is semi-dwarf bread wheat variety. In maize, DMR New Delhi or IIMR Ludhiana, now it is shifted. Pratap Composite Mukta 4, Pusa Early Hybrid 5, Pragati Deccan Hybrid 115, Pro 345, PEMH 5. In sorghum, Pusa Chari 6, Chickpea BG series BG6108, that is Kabuli kind, and BGD 128. Pigeon pea cultivar Pusa 2001. Then in Basika, Pusa Tarak Karan Rai. In soybean, DS 9814, that is yellow mosaic virus resistant. In bottle gourd, Pusa Santrushti. In vegetable cowpea, Pusa Sukmal. Tomato processing, the important varieties are DT10, Pusa 120, Pusa Gaurav. Cabbage hybrid, KGMR 1. That is the first variety by public sector in cabbage. Sape seed moisture percent for vegetable seed storage is 4 to 6%. Artificial seed is developed by Morishige. Karancal helps in germination. Operculum helps in impervious to water and inhibition of germination. These are structures on seed. Different structures are coracle and operculum. Coracle helps in germination, whereas operculum inhibits germination. High lysine content mutants of maize are opaque to different series of opaque are there, then Shakti, barley, hiplori. Coming to the special treatments to break the dormancy. Pre-chilling treatment is practiced in onion, lentil, rapeseed, linseed, cherry, lucerne, lettuce, broad bean and radish. Pre-chill and KNO3 treatment is given to mustard and rapeseed, cabbage, cauliflower. Preheat at 30 to 35 degrees Celsius and pre-chill and GA3 treatment to oat, barley and wheat. Removing shell and preheat 40 degrees Celsius to groundnut. Pre-wash in sugar beet. KNO3 treatment to all the solanaceous crops like chili, tomato, cabbage, tobacco. Then preheat treatment 50 degrees Celsius to rice. Coming to the planting, general planting ratio of for hybrid seed production. In case of wheat, rice, oat, barley, 8 is to 2 is generally practiced. In bajra, 3 is to 1, castor, 3 is to 1, cotton, 4 is to 1, sunflower, 3 is to 1 or 15 is to 5, okra, 2 is to 1, maize, 3 is to 1 or 4 is to 2, red gram, 6 is to 1, brinjal, 3 is to 1, then safflower, 3 is to 1. Sugarcane, medicinal plants and flower plants are not covered under Seed Act. This point you should remember. Sugarcane, medicinal plants and flower plants are not covered under Seed Act. Important terminologies. Nicking is synchronization of flowering. Polyembryony is discovered by Leeuwenhoek. Coming to the utility of male sterility in different crops. CGMS is generally practiced in sorghum, bajra and rice. GMS in rice and pigeon pea. CMS in onion, vegetatively propagated plants. CMS reported first in Onion by Jones and Davis. Coming to the adventative embryony. This is the condition where embryo arises from maternal sporophytic tissue, that is by nucellus or integuments. These are the forms of apomixis. Remember, true polyembryony where more than one embryo arises from single embryo sac. False polyembryony is more than one embryo arises from more than one embryo sac. Citrus, mango are polyembryony from nucellus. Coming to haploid parthenogenesis and diploid parthenogenesis. Just give me the definition of parthenogenesis in comment section. I just want to see whether you are attentive to the lecture or not. Haploid parthenogenesis examples are Nicotiana and maize. Diploid parthenogenesis examples are grasses. Debearder is removal of awns or appendages. Example, this is practiced in barley and oat. The formation of 7 cell and 8 nuclei, how it will take place. Each microspore mother cell will, this is for female part, where megaspore mother cell will get into one megaspore, where megasporogenesis takes place. After that, megagametogenesis process happens, where female gametophyte is produced, and that is 7 celled and 8 nuclei. Similarly, for male part, each microspore mother cells will undergo division, that is microsporogenesis. 4 microspores are produced. Microgametogenesis is the next process through which 4 male gametophytes are produced, in which 2 generative and 1 tube or vegetative nucleus are produced. Largest cell inside embryo sac is polar nuclei. Multiple fruit or cirrhosis. A fruit that develops from a fusion of carpels from more than one flower. Example, jackfruit, mulberry, pineapple. These are all cirrhosis kind or multiple fruit. Aggregate fruit means several ovaries or several carpels of single flower fused to form a fruit. Example, strawberry, raspberry, etc. Fleshy fruits. Berry type are grape, tomato, banana. Pepper or squash, cucumber, melon, gourds. Drupe type are mango and coconut. Newsletter of AOSCA published quarterly. Sample drawn from government department are service sample. Sample drawn from by seed inspector are official samples. Sample drawn by farmer for volunteer certification is charged sample. This is also very important. The type of physical purity parameters that can be separated by blowing is other crop seeds. Seed lot size of maize is 40,000 kg. These are all bits taken from different books. Okay. Seedlings with sufficient lateral roots but lack in primary roots is normal seedlings. Seeds that do not imbibe water are hard seeds. Seeds that imbibe water but don't germinate are fresh ungerminated seeds. Seed testing in pelleted seeds is depelleting. GADA test is practiced in monocots. Iron chloride test for to test the injury in seeds. Seed crushing test in jute. TZ test to see the viability and vigor. Radiography is done in woody plants. Hard seed coat is seed in Leguminosae and Malvaceae. In beans, stophiol plug prevent embryo of water, thereby prevents germination. In Malvaceae, chalazal cap at chalazal end prevents entry of water. This also you should remember 2 points. In case of bean, stophiol plug that prevents entry of the water into embryo so that it prevents germination. In Malvaceae, chalazal cap that prevent entry of water into chalaza. Perisperm is the storage organ in onion, that is also observed in pepper, coffee, lychee, etc. Naturally occurring inhibitors are dormins. that is by ABM. Diseases specified for seed certification are known as designated diseases. Notification of variety or crop is done by Central Seed Committee. Coming to different sections under Seed Act 1966, till now I have discussed this many a times, but in systematic way I have not told from section 1 to section 25. So one by one we shall see. Section 1 talks about short, little, extent and commercial terms of seed. Okay, section 2 talks about definition of seed. Section 3 Central Seed Committee. Section 4 Central and State Seed Laboratory. Section 5 power to notify kind or variety. Section 6 minimum limits of purity and germination etc. Standards. Seed labeling. Section 7 talks about regulation of sale of seed. Section 8 about certification agency. Section 9 certification issues or grant. Section 10 talks about revocation of certificate. Section 12 talks about seed analysts. Section 30 talks about seed inspector. Section 14 about power of seed inspector. Section 15 about procedure to be followed by seed inspectors. Section 16 about report of seed analyst. Section 17 about restriction on import and export of seeds. Section 19 talks about penalty. Section 25 about power to make rules. These are the important sections from 1 to 25. Now, apigeal germination is common in dicots except peas and broad bean. Chemical test for both vigor and viability is TZ and GADA. Protection of plant varieties of field crops is 15 years and for trees and shrubs is 18 years. As per ISTA, number of seeds taken for germination test 100 in 4 replication for small seeds and 50 in 8 replications for bold seeds. Coming to the mode of pollination, in rice we find self-pollination and generally chasmogamy. In wheat, oat, barley, groundnut as well as rice, self-pollination and cleistogamy. Cleistogamy is closed flower condition, whereas chasmogamy is opening of the flower after fertilization, okay, or pollination. In case of maize, carrot, sorghum, sesamum, safflower, sunflower, protandry is observed. Bajra, sugarcane, nizer, castor, protogyny. Alfalfa, vanilla, hercogamy or mechanical barrier is found. Brinjal and linseed, heterostyle. Self-pollination is observed in soybean, black gram, green gram, lentil, chickpea, cluster bean, cowpea, linseed, tomato and methi. Cross-pollination is found in maize, mustard, rapeseed, sunflower, cucurbits, radish, sugar beet, palak. Often cross-pollination is found in cotton, sorghum, red gram, brinjal, okra, sesamum, castor and safflower. Self-pollination, that is less than 5% of cross-pollination is found in paddy, oats, wheat, ragi, barley, black gram, green gram, bengal gram, soybean, jute and groundnut. In case of vegetables, cowpea, cluster bean, tomato, lettuce, tobacco, that is a photoblastic plant, garden pea and French bean. Cross-pollination, that is more than 50%, found in field crops like maize, bajra, sunflower, nizer, castor, mustard, sun hemp, sugarcane. In case of vegetables, cabbage, carrot, radish, amaranthus, cucurbits and onion. Often cross-pollination, where 5 to 30% of cross-pollination is found in sorghum, red gram, sesamum, cotton, lucerne, berseem, safflower and triticale. In vegetables, bhindi, brinjal, chili, bell pepper and sweet pepper. These all things we have already discussed, like the objectionable weed and all. Now, the notified variety or farmer's variety that is in public domain is called extant variety. Seed years are 1961 and 1993. Nomograph useful to predict the seed viability period, which was given by Roberts, and he wrote a book called Seed Viability. Ideal temperature for bolting in cabbage is 4 to 7 degrees Celsius. An introduction to seed technology was written by Thompson J.R. National Seed Research and Training Center is located at Varanasi. Seed research published by ISST. Chemical used to induce femaleness in cucumber is silver nitrate, that is AgNO3. Working principle of indent cylinder is based on seed length. Seed Science and Technology, the magazine published by ISTA, and Seed Technology by AOSCA. Pearson's square method can be employed in seed blending. In cereals, major portion of protein is present in aleurone layer. Sensitive species for testing the toxicity of germination paper is in mustard and onion. Sunflower will have dormancy of 45 days after harvest. That means we cannot sow it immediately. We can sow it after 45 days of harvest. Coming to the determination of moisture content, low constant temperature oven 103 degrees Celsius plus or minus 2 degrees Celsius for 17 plus or minus 1 hour is low constant temperature oven, whereas high constant temperature oven is 130 degrees plus or minus 2 degrees Celsius for 1 to 4 hours. Rain favors pollination in pepper. For maintenance of variety, positive selection is done. For production of foundation and certified seeds, negative selection is done. Intercropping is allowed in certified seed production of oilseeds and pulses, not in cereals. In sunflower seed production, multiple heads is generally found in R line. Viability of dormant seed can be detected by TTC test. As per ISTA, the germination test has to be conducted with pure seed fraction. Cartagena Protocol imports of transgenic it talks about. ISTA motto is uniformity in seed testing. Natural germination inhibitor found in seed is coumarin and abscisic acid. Minimum temperature for wheat germination is 20 degrees Celsius. Salt tolerant variety of basmati rice released by CSSRI kernel is CSR 30. Field cloned buffalo is Garima. Its calf is Mahima. 2012 is declared as the year of horticulture. As per 9th Agri Census in 2010 and 11, average operational holding is 1.16 hectare. These are all outdated information. We won't cover this. Rye is a cross pollinated crop. Then the term double fertilization was given by Navashin and Grignard. Male sterility by Colreuter. Syngamy, it's a process of fertilization. Thalamus is found in apple, coconut, and plum have endocarp, whereas mango and guava, the edible part, is mesocarp. Different conditions. Parthenocarpy is found in banana. Multigerm is found in sugar beet. Vernalization is for flower induction. Viviparous condition is found in jackfruit, that is germination on tree only. Then devoid of endosperm is found in Orchidaceae or orchids. Smallest flower is Ravolfia microscopia. Largest seed is Laudacia maldivica. Largest flower is Ravolfia species. Smallest seed is orchids. Different terms used: nipping in cotton, curing in tobacco, wrapping in sugarcane, stripping in jute, propping in banana. Ethylene treatment is done for fruit ripening. Glyphosine is sugarcane ripener. Gibberellins are used to increase the fruit size. NAA is used to increase the number of flowers. This is the specific purpose of different chemicals. Different deficiencies. White bud of maize is due to zinc deficiency. Black heart of potato due to oxygen. Fruit cracking of tomato due to boron. Mango malformation by NAA deficiency. Different conditions like brinjal is having heterostyly, means different style length. Cotton delinting is generally done. Then in castor, endospermic seed condition is found. Sugar beet, multigem, radish, steckling. Different hypothesis given. Davenport gave dominance hypothesis. Shell and East gave over dominance. Mather, disruptive selection. Accelerated aging test is done in soybean. Radical emergence test in maize. Conductivity test in Gordon pea. Controlled deterioration test in rapeseed and mustard. Then ploidy level test in seedless watermelon. Next, some of the important points from Vasudevan and Satish and co-workers book. Optimum temperature for vernalization is 1 to 7 degrees Celsius. Rye, sugar beet, cotton, carrot require vernalization for flower initiation. Phenomena of repeated exposure to low night temperature separated by periods of high temperature for floral induction is thermoperiodism. Vernalization is flower initiation by decreased or low temperature treatment. Conversion from PFR to PR takes place in dark. This concept I have already explained in first video. Flowers without corolla are opetalous. Example in Annona. Position and arrangement of calyx and corolla in flower is estivation. Palynology is study of pollen. Monoadelphous examples are malvaceous crops. Diadelphous, Fabaceae. Polyadelphous in castor. Cryptoantherus is anthers hidden inside corolla tube. Parentherus is a condition where anther wall exerted over flowers. Synandrous is a condition where filament and anther are completely fused. Example in cucurbits. Multicolor ovary is example are bhindi cotton found in Malvaceae. Some flower are hermaphrodite and some pistillate found in gynodioecious condition. Some flower are hermaphrodite and some staminate in androecious. Staminate means male part and pistillate is female. Here androecious and gynodioecious condition is the term given. Then andromonoecious is hermaphrodite flowers plus staminate means male part. Gynomonoecious is hermaphrodite flowers as well as female or pistillate flower conditions are found. Next, runners, stolons and suckers are found in subaerial stems. Apomeosis is the process of production of unreduced embryo sacs. In this, apospory and diplospory are the two conditions. These are different kinds of apomixis only. Apospory and diplospory. In diplospory only parthenogenesis, pseudogamy, this condition will come. Pseudogamy is nothing but pollination is necessary for embryo development, but fertilization of egg cell does not take place, whereas fertilization of secondary nucleus occurs due to development of endosperm. Thin wall enclosing haploid egg cell in embryo sac is follicle. Genetic consequences of geitonogamy are same as autogamy. Allogamy is also known as xenogamy. Wheat, barley, oat have cleistogamy. Rice has chasmogamy. Dicliny condition is again divided into monoecious and dioecious. In monoecious, male and female on same plant, and dioecious on different plant. Diacliny is a unique sexual condition. Now, some of the examples for pseudocarpic fruits: sandbar and burdock. Multiple fruits: pineapple and mulberry. Aggregate fruit: strawberry, raspberry, blackberry. Dehiscent fruit are splitting along the built line of pod or fruit. Simple fruits are divided into fleshy fruits and dry fruits. Dry fruits are again divided into dry dehiscent fruits and dry indehiscent fruit. Dehiscent means legumes, follicle, capsule, calyx. Then dry indehiscent fruit means caryopsis, achene, nuts, schizocarp, etc. Treatment with ABA induce desiccation tolerant somatic embryo. Hydrated synthetic seeds are produced in recalcitrant species. Cotyledons are nothing but seed leaves. Hypocotyl is stem like embryonic axis below cotyledon. Radicle is embryonic root. Two-thirds of the embryo genome is of maternal origin. In mature seeds, endosperm is almost degraded and embryo is covered by testa. Awns arise from tip of the seed. Elaiosomes are fleshy appendages rich in lipids and proteins. They aid in seed dispersal by ants. Means elaiosomes are having oil content, and these are attractant to ants and help in pollen dispersal or seed dispersal, whatever it may be. Wings on seeds are present in drumstick or moringa seeds. Albuminous means soluble in water. Albumins are soluble in water. Globulins soluble in saline. Prolamins soluble in alcohol. Glutalins soluble in alkali. Sterols are soluble in alcohol. I repeat, albumins are soluble in water. Globulins in saline condition. Prolamins in alcohol. Glutalins in alkali, and sterols in alcohol. Proteins of wheat are glutenin, paddy is oxygenin or glutenin, barley is hirudin or hordin. Prolamin in wheat is gliadin, maize zine, paddy lacks prolamin. Legume proteins are vicillin and legumin. Dicots absent in glutelins. In gymnosperm seeds, food is stored in megagametophyte. Protein of peanut is arachidin and conarachidin. In soybean, glycine, hemp, gliadin, peas, vicillin. Seed coat impermeability in Sida spinosa due to lignins. Allelopathic chemical is in eucalyptus is terpenes. Non-living bodies of cell are collectively called cell inclusions. Mechanical tearing of abscission tissue during seed dispersal. The phenomena is called schizolysis, that is mechanical tearing of abscission tissue. Fatty acids are present in lysosomes: erucic acid, castor ricinoleic acid. C source for synthesis of membrane lipid is sucrose. In castor, Calvin cycle enzyme phenol in chloroplast. Carotenoids contain oxygen. Carotenoids without oxygen is found in, that is keratin. The carotenoid which contains oxygen example is xanthophyll. Origin of rice is glutalin, legumin of pea is globulin, and trypsin is present in soybeans. Waxy corn has gummy starch. Popcorn has hard starch. Flour corn has soft starch. Friends, this is all about part 5. We'll discuss the remaining in part 6.